So, Bismillahir Rahmanir Rahim. We are going to talk about differential gene expression and how it contributes to development, or in other ways, we can phrase the same question: how during development different cell fates are established, how cell lineages are established due to the differential gene expression. So we learned yesterday that after fertilization, which is fusion of sperm and egg, we have a diploid organism which undergoes successive mitosis. The same cell divides repeatedly, which we refer to as cleavage, cell divisions. And then after passing through blastocyst stage and eventually the gastrulation, what we see actually is different cell types. So in first 21 days or so, first 3 to 4 weeks, what happens that we have from single cell, which contained 46 chromosomes, a fertilized egg, we have different kinds of cells, different types of cells, they emerge because there are a couple of hundred cells or a couple of thousand cells after successive cell divisions. And we see determination of cell fates, or cell fate determination, where we will see, let's say, future liver cells or skin cells, heart cells, and so on, different kinds of cells. Now, interesting thing is, each of this cell type which gets determined early on, they all contain 46 chromosomes each. They all inherit these 46 chromosomes from their progenitors, which are descendants of this particular cell type, which is a fertilized egg. When you have 46 chromosomes in each cell type, question arises: when same gene set is present here, each cell type contains same set of genes, how can they be different from one another? Okay. And that's where the key concept of differential gene expression comes in. Differential gene expression means, this cell will be expressing key cells which are responsible for example, neurons. This cell will be expressing cell types which are going to be responsible for kidney fate, or liver, or skin, or heart, and blood. Within blood, you will have white blood cells, platelets, etc. But before such differential gene expression kicks off, you have to go through specific pattern formation and the process of morphogenesis, where the cells are organized in a three-dimensional structure. And all this is happening in these 3 to 4 weeks. And eventually we see, as a consequence of differential gene expression, we see organogenesis, or different organs start becoming visible. A very interesting experiment was done nearly 30 years ago, where they showed that, normally in sexually reproducing organism, what we have sperm cells and egg cells. They both are haploids. Okay, they both are product of meiosis. They both fuse and result in a diploid, and this fertilized egg, which is now diploid, it has the potential It has the potential to generate all different kinds of cell types which are required in a multicellular organism, and then eventually it results in development of a completely normal individual. This concept is called totipotency, when a cell type has the potential to give rise to development of a completely normal individual. This was known in plants, this was known in some other species, but in case of higher eukaryotes, for example, in sheep, what they did, they showed this concept. What they showed, they said, okay, we are going to take a skin cell from the udder, this part, which contains the mammary glands in case of sheep. So they took a diploid cell, the skin cell here. What they did, they took nucleus of this diploid cell, the 2N nucleus, which is containing the diploid set of chromosomes, and then what they did, they took an egg cell from another sheep, and this egg cell was enucleated. Its nucleus was removed, and the diploid nucleus from the udder cell was fused and transplanted within the egg cytoplasm. So this is the egg cytoplasm, and now the nucleus is of a diploid cell. It means this egg never went through fertilization. This underwent cell divisions, and then it was implanted or transplanted within a female foster mother. And what they could show, that this egg, which contained diploid nucleus of a fully differentiated skin cell, resulted in birth of the sheep, which was called Dolly. Now, what we learned from this interesting experiment is that the diploid nucleus of a fully differentiated skin cell, it can be reprogrammed. It means the diploid 2N chromosomes in a fully differentiated cell, it does not undergo irreversible changes. It contains all the genes, all the chromosomes. All you need to do is to reprogram from this nucleus, from the skin program, back to early dedifferentiated state, and then the egg cytoplasm, it supports all its development to complete organism. It means if we are able to reprogram a fully differentiated cell, a diploid cell which has undergone differentiation can be totipotent. However, a key message, a key learning point which you should be paying attention to is that from this stage of an egg in a normal reproduction system where sperm fertilizes, from here going all the way to adult, and this udder cell or skin cell from head or wherever, it went through a gradual developmental program where its nucleus went through differential gene expression or certain programming, where certain genes which are specific, which are responsible for development of this particular feat, they are switched on, and all the others, for example, eye specific, leg, heart, liver, blah, blah, they are switched off. That's why these cells became part of the udder. And this program, after differentiation, in a normal body is irreversible. Here it is not totipotent. Only when you take it out, isolate its nucleus, transplant it into the egg cytoplasm. The second major message is that this is reprogrammable, but by the cytoplasm of the egg. It means cytoplasm of egg contains something very essential information which can push this through reprogramming. It forgets all the gene expression program of udder or skins and now reprogrammed to become a nascent, a naive, a novice, a baby nucleus which can go into completely new developmental program. So we are going to learn today journey from normal fertilized egg to differentiation, how different cell types are developed due to differential gene expression. This is how our life starts. On the day one, we are like, 1 day old zygote. When sperm fuses egg, this is called zygote. When zygote undergoes cleavage divisions, okay, and then comes a stage we start calling it an embryo. Okay, here if you pay attention, 1 day old embryo to 3 days, 4 days, 15 to 17 days, and 18 to 19 days. Now here you see slight change in the shape, and eventually this is the stage where organogenesis, neurulation has started, and eventually you see different tissues and organs emerging. Now these first 3 weeks are very crucial, where cell fates are being determined. सेल्स के मुकद्दर का फैसला हो रहा है कि तुमने मुस्तकबिल में आंख बनानी है, स्किन बनानी है, दिमाग में न्यूरॉन बनानी है, दिल बनाना है, शरेयान बनानी है, आर्टरीज बनानी है, वेन्स बनानी है. You name the kinds of cell types we contain in our body. So all this is being decided in first 3 weeks. Now, when all this is taking place, in order to simplify the development, or in order to understand this process, we divide into different phases. So we say, for example, cell fate determination. Okay. Morphogenesis. So I come later on this one. So cell fate determination is, when fates of different future cell types are being determined or established. So you have, for example, different cells. Imagine these are a couple of 100 cells, and their fates are determined. These are future. These 3 are determined or established to be I. These 3 are determined to be heart. These 3 are determined to be, brain cells. And then, of course, they divide. They will keep dividing mitotically. Okay. There comes a time when these cells, they have to slowly differentiate now into their shape, start slowly becoming different. Their region within the embryo, after certain stages of mitotic division, start becoming different. These 3 led to, for example, these cell types, future. Its progeny of these 3 turns out to be, within the embryo in a specific region, they start becoming founding heart and then brain. Now, these fates are determined after every mitosis of these cells, from here all the way to here. The genes which are switched on in I-specific, let's call them I-specific genes. These genes through every mitosis will remain on. And the ones which are non-essential for eye development, which are dangerous, let's say heart-specific genes, they will be off in eye, and vice versa. The eye-specific genes will be off in heart, and heart-specific genes will be on. When a cell divides into two, 2 to 4, 4 to 8, so successive mitosis ensures that these cell type specific gene expression program, we call this cell type specific genes, they are maintained. Now the word is, after establishment, they are to be maintained in the on state. And then comes a stage where we see very clear kind of eyes, where will be eyes somewhere here. Okay, some structure starts appearing, which may not look like adult eyes, which we are born with, but this eye is the precursor or founder of this. This stage is called differentiation. So from self-fate determination or establishment of self-fate to differentiation, there is a time before tissues become clearly distinct, organs differentiate. For example, my skin is now, after differentiation, there's a stage, we call it terminal differentiation. Terminal differentiation is the stage when it has attained the ultimate destiny. It has exited the cell cycle. It is no more dividing mitotically. It has attained the ultimate destiny, which was established. So from self-fate determination to differentiation, there is a time lag, but of course, the determined self-fates, they are maintained generation after generation during mitotic division. Along the same stages when this development is taking place, morphogenesis takes place. Morphogenesis is basically organization of cells in a specific order, in a specific organization, in a specific three-dimensional organization. And I gave you an example of morphogenesis yesterday, that early on embryo here, for example, is a two-dimensional structure, and after gastrulation, so gastrulation is already a three-dimensional structure where you have ectoderm, mesoderm, and endoderm. These cells further undergo organization and after pattern formation, where eventually you see organized organs developing. So when cells are being organized in these 3 weeks in a specific pattern, which attains three-dimensional structures, which results eventually, and it is due to morphogenesis that we see very distinct structure of arms different from legs, not at this stage. Remember, this is ultimate result of morphogenesis, which took place here in first 3 to 4 weeks. Okay, and then eventually, of course, we grow, which is called the growth phase. We yesterday covered in detail all this life cycle. This is just a recap that we are going to go through this early development, which is only up to here, and then look at the differential gene expression. Who tells or how, let's say X number of chromosomes, these two cells also contain X number of chromosomes, or let's say X is confusing, N number of chromosomes, 2N, 2N. Each of these 8 cells contain same 2N, same set of chromosomes, but eventually they start becoming different. At what stage and how differential gene expression is kicked off, that's what we are going to learn. Yesterday, I gave you an example about Xenopus, and I showed you that in Xenopus, you know, animal and vegetal pole already comes in, different polarity, where at vegetal pole you have a protein which after fertilization due to rotation uh, of animal pole uh, moves near the gray crescent, and there is degradation of there's uh, degradation of GSK, and wherever GSK gets active, beta catenin is degraded, and where these uh, proteins are present, they protect the beta catenin, and you have clearly uh, two different regions within same cytoplasm. This is a cartoon which is showing you more or less a picture in which, let's say, cytoplasm contains some specific contents within the cell. After fertilization, what you see, you have a cell division where the contents of cytoplasm are divided equally into two cells. So we call this kind of cell division symmetrical cell division, where one cell divides into two cells, and both the resultant cells are containing same content, same number of chromosomes, and then of course the cytoplasmic content. Early on during development, due to cell-to-cell communication, due to environment, so early on we still cannot define, we still cannot understand. We know signals, but how these signals are induced in our mother's womb, how cell knows I have to divide now asymmetrically instead of symmetrical division. I have to divide asymmetrically and result in 4 cell types where 2 will look alike and the other 2 will look alike. These 2 cells may again divide asymmetrically, and one cell will look similar and the other one will be different, and this will divide asymmetrically. One cell will look like this one, and the other one will look like this cell. Okay, so this kind of asymmetrical cell division due to cell-to-cell communication or signal within the embryo as well as coming from mother, we see such kind of distinction taking place. Now, what are the consequences of such asymmetrical cell division? Look, such asymmetrical cell division will have indeed consequences because you have, for example, this one has cytoplasmic content which will feed or send a signal to nucleus, activate certain processes which this cell cannot do. Although this cell looks at an advantage to activate certain process, but this cell is also at an advantage because this does not want to activate the process which is active here. This is going on a different route than this one. This cells from this one will attain different identity or fate as compared to this one. Okay, so this is a cartoon which is showing that we have a cell, and this cell is producing some signals. These are those signals. It can be a protein, it can be some amino acid, it can be simply a chemical. Okay, it can be simply calcium. Okay, whatever signal it is, based on its nature, it can diffuse into a cell, and this cell which has received this particular inducer, it then activates certain genes within this within the nucleus of this gene, and this particular cell which has received this, look, this signal is being received by on the cell membrane. This is shown a receptor. okay, where this signal is fine. The signal diffuses or gets transduced. Ultimately, within the nucleus, a transcription factor gets active. This transcription factor, it binds to the DNA and activates a certain set of genes whose protein is made. However, another neighboring cell, which does not receive the signal very nicely or perceives it nicely, look, its receptor is still unbound, it did not perceive this signal in a positive manner. So the transcription factor, which was active or moved into the nucleus due to this particular signal, this transcription factor does not move in, or certain set of genes are not on here. So the changes which took place in this cell, they did not take place in this particular cell type. So the gene, let's say gene A is on here, but gene A is off there. If it is taking place in the nucleus, such a signal will not just touch one gene, but it may touch a couple of 200 genes. 200 genes are on only characteristically in this cell type, and they remained off here. What is the consequence now? You have two different populations of cell. When mitotically these cells will divide, it will contain these 200 key genes on, and this will have 200 genes off. But this particular cell will have certain genes on, which this one doesn't. So you have a distinction coming in. How these signals can impact development when you have many cells. Here we just showed you two cells. Is it clear so far? Is it clear so far?
Yes, sir.
Yes, sir.
Do you still see my screen, or screen is gone?
We can see your screen.
Good. So let's look at a growing embryo and try to see a picture which may emerge as a 3D picture, although on the page it looks a two-dimensional structure. Look, here we have cells organized, in a very nicely manner, closely attached cells. After cleavage divisions, you have mitosis. Now, what happens? Imagine you have such a signal being produced by a specific cell. Now, imagine this cell, this signal producing cell is somewhere here, where a lot of signal is being produced. Or maybe within the cytoplasm of a fertilized egg, these signals are present. But imagine after some cell divisions, we have such a signaling going on. Now, this line is showing you that this signal is how strong this signal is here as compared to here. On the x-axis, if we say this is the point, source of signal, the maximum signal will be here. And the minimum signal, because this is a diffusible signal. Such signals in development are called paracrine signals, which can be, which are diffusible. Okay. Now, this cell, the one which is here, let's say it's receiving 100 molecules because this is close by, and this one is receiving 10 molecules of this signal. This one maybe is receiving 80, 60, 40, 50, whatever. I mean, don't think it's so precise, but we can look at this and we can say, the gradient. Now, this is a gradient, high to low gradient. In development, we call such gradients are called morphogen gradients, because they are going to play a role in morphogenesis. They are going to activate certain changes within the cell. So this signal perceived by this or this one, when we have this level of concentration of morphogen, let's say first two cells fall in this concentration gradient. These two are under this much concentration gradient. This is going to activate a certain gene cascade, certain key genes, which are actually responding to this threshold concentration gradient of morphogen. The next two are activating certain developmental gene expression program, and look at the fate. These two are going to attain a different fate as compared to these one, and the one which receive least concentration, they activate or switch off or switch on different genes. So what we learn, that the position of each gene within the embryo is very crucial, and the fate or key developmental genes which are going to be switched on is going to respond to specific concentration of the morphogen due to its specific position within the embryo. So here you can generate a French flag, for example, blue, red, and red, blue, white, and red. So this white destiny is due to certain level of this from here, I would say this concentration gradient and this concentration gradient of morphogen, the signal activated only certain set of genes. This is actually the onset of, I would say, kickoff differential gene expression. Now, why differential? We have, for example, 46 chromosomes in this cell, 46 in this one, 46 in this. When each cell contains same 46 chromosomes, which means same set of genes here, same set of genes here, each cell contains same set of genes. However, due to this signal, this morphogen gradient, cells here have switched on certain kind of genes which are off here, but white ones have switched on other set of genes which are off here, and vice versa. So this is selectively switching on, switching off genes is called differential gene expression. In other words, we also call it cell type specific gene expression program. Let's look at an example which will make it more clear, because this is a real example which you can very clearly correlate. And this example is about how digits in our hands are developed. For example, this is so the thumb, first the index finger, second, third, and the smallest finger. This is the point. This particular point is the limb bud. Limb bud is just a little projection in early developmental stage here. Here already cell fate might be established, but at this stage, we don't see it digits. We don't see these fingers, hand, and elbow. Just few cells are determined early on, and here comes a signal, a morphogen, which we call sonic hedgehog. The morphogen is sonic hedgehog, which diffuses. This is the point where maximum signal of sonic hedgehog is. It diffuses, and you can draw a gradient like this. You can say This is the highest and this is the lowest after diffusion. The cells which are going to be exposed to lowest amount of sonic hedgehog as compared to cells here, as compared to cells here, different genes are going to be expressed. So the little finger is formed when you have maximum concentration of sonic hedgehog in these cells. These cells which contain intermediate signal is going to activate certain cells. These genes are called homeotic genes, or in mammals we call them Hox genes. These are, the master regulators of development. We will talk about them in later part of the lecture. Master regulators. They are so crucial genes, ladies and gentlemen. If one gene, just one gene out of a set of these 8 genes is misregulated, we may lose our eyes, we may lose our hand, we may die. Just a single gene. That's why they're called master regulators of development. They are differentially expressed, and look, due to highest concentration of sonic hedgehog, cells which were here, eventually after mitosis, eventually when they start differentiating, the cells here already were determined. Okay, gentlemen, you are going to make little finger. Okay, you take this route, divide and pass on information to all your future mitotic progenitors, and eventually, because specific genes will be switched on, it should develop into smallest digit, little finger. And the thumb should be these cells. Gentlemen, you are exposed to smallest amount of sonic hedgehog. You pass on the information to all your mitotic progenitors, and eventually you develop as thumb. And this gradient, just like French flag model, you have such a beautiful structure. Do you see symmetry? And look at the symmetry. Have you ever observed yourself closely? If you haven't, stand in front of the mirror and appreciate whoever has made you. Appreciate how beautiful he, she, or Mother Nature, whatever you call this, you are made. Flawless. You cut yourself from, have a ruler here, and you have a bilateral symmetry. Even the eyebrows, your eyelashes, I mean, you name it. You have nostrils. एक nostril बंद हो जाए तो वक्ता पड़ जाता है बंदे को, रात को सांस नहीं प्रॉपर आती. So all this is due to early on self-fate determination due to these morphogen gradients. The process of development was, we owe a lot to Xenopus, to sea urchin, but I love the discoveries in fruit flies, because I think what was discovered in fruit flies, it changed the way we used to look at development. Although this is an invertebrate and we humans are vertebrates, okay, we look very different. It has 3 pairs of legs, antennas, eyes, thorax. So its body is divided into 3 major parts: head region, thorax, and abdomen. And the body is segmented. Okay. Now, people who are interested in understanding that process of development, how we develop, they said, let's look at how flies develop. Looks very crazy question. We are interested in human development. but we are studying it in flies. And we will appreciate after this lecture what fly contributed to in the lessons it taught us. Fly completes its life cycle in 10 days. I told you this in genetics lecture when I explained you the Morgans and discoveries. It lays an egg after fertilization. This egg is a very specialized cell because, you know, after fertilization, the cell divides into two. Not cell, the nucleus divides into two. These two keep dividing, 4, 8, but the cytoplasm remains the same. There's no cytoplasmic division, only the nucleus divides. After 24 hours, you know, from going from here all the way to here, एक सुंडी, एक caterpillar, एक larva develop होता है, which we call it's called first instar larva. Another 24 hours, it grows a bit, second instar. Another 24 hours, third instar, and another nearly 3 days, larva undergoes metamorphosis and pupa emerges. And in 10 days, after 3-4 days, from egg to the adult, it's total 10 days. From pupa to adult, it's like 3 to 4 days. Okay. However, like humans,
can you zoom in a bit? They cannot properly see the diagrams. They're asking if you can zoom in a bit.
How can I zoom in?
There will be this plus sign if you, or control plus करके देखें अगर उससे zoom in होता.
Yeah. Can you see now?
Yes. Now, is it better, Uswa?
Yes, thank you, sir.
Thank you, Abdullah. I don't know what I will do without you, man. So, look, now is it clear? तो अब ये क्या हो गया? अब मेरा pointer कहाँ गया? उन्हें भी मैंने दसो. Anyway, so what I wanted to tell you from here all the way to here, like in us, in humans, the first 3 weeks are important. In embryos of flies, first 10 to 12 hours are so crucial. The morphogenesis, the gastrulation, all this is taking place here. These nuclei, when they divide without the division of the cytoplasm, they all get organized, you know, within all these nuclei, they divide at the same time and they again start DNA replication. Again, they divide, again start DNA replication. They are synchronous. There's synchrony. They all replicate, divide at the same time. Okay. While they all share the same cytoplasm, we call this stage syncytial blastoderm. And then comes a time when all these nuclei move towards periphery of the embryo here. And this is the first sign where you see small grooves coming in here. And these small grooves is the first sign that these nuclei are just going to become individual cells. And this is called when, you know, you see cell membrane emerging from this one big cell membrane of cytoplasm. There's like a waterfall effect. You see, you know, cell membrane going down and each nucleus is encircled in its individual cell membrane. We call this cellular blastoderm. Slightly later at this stage, what happens, I mean, the cells keep dividing mitotically now, but there is already early on, much before cellular blastoderm stage, early on, we have morphogens which are deposited by mother already here in the cytoplasm. After fertilization, when egg is laid, before the process of fertilization, mother prepares the egg in such a way, we call them maternal genes. Mother deposits them here because mother knows my baby is going on a long, long journey and it needs food, it needs, you know, proper developmental program. As soon as fertilization takes place, these morphogens get activated, and we see that morphogenetic effect we talked about, you know, this French flag model, etc. That starts here already. Now what happens at stage 13, which is like first few hours, I think 3 to 4 hours maybe, we see embryo becoming segmented. The 14 segments. So I said there are 3 major parts of fly body: head, thorax, and abdomen. They rely on these 14 parasegments. And these parasegments, these 3 are 1, 2, 3. Do you see this hand which is on the slide? These 3 segments.
Yes, sir.
These 3 segments, they are basically the head region. They are going to make head. Okay. Next 3 segments. Next 3.
Sir, how is that determined?
Yeah.
That the 3.
Yeah, we will talk about this. Next 3 segments, these ones, are going to be thorax, which is this region. And then last 8 segments, parasegments, these are called para, they are going to be future abdomen. Okay. So as she asked how they are determined, this is what we are going to learn today. Okay. The morphogen gradients, which I just gave you an example here, this kicks off, this is actually learned from fly system. We came to this French flag model, this morphogen gradient model from fly development. So partially I'm giving answer to you that, you know, these 3 cells, they have received a signal, activated certain sets of genes, which are determined to make future head. Okay. Now, detailed answer is going to start now. As I said, the mother deposits certain products of genes. Within the ovary, let's say this is the ovary of the fruit fly female. Within the ovary, you have, you know, these egg development is taking place. This is the future egg cell. These cells, these are all diploid. No, these are polyploids. These are not diploids. Polyploid. Polyploid means they are, instead of 2n, they may be 8n or 16n, whatever. Diploid cells in ovary are here, okay, which are called the follicle cells. These are 2n's. These are polyploids. So these polyploid cells, they deposit tons of messenger RNAs of key maternal genes. And these messenger RNAs, when this egg is laid, as these messenger RNAs are deposited at very, very specialized positions or specific position. For example, Bicoid is one of the messenger RNA. This is the maternal effect gene, or maternal gene, we call this. Mother deposits this. And similarly, on the posterior side of embryo, there are other maternal effect genes, for example, nanos, caudal, Oscar. Okay. Here you have Bicoid, there are other hunchback, blah, blah, blah. As soon as the fertilization takes place, sperm comes in, it hits the embryo, drops the nucleus inside, the egg nucleus and the sperm nucleus fuses, the fertilization takes place, it becomes a single cell. One cell divides into 2, 2 to 4, 4 to 8. As soon as fertilization takes place, the first thing what happens is these messenger RNAs on the anterior side or posterior side, they are translated. their messenger RNAs are translated, and now protein products are there. Remember I told you fly embryo is a syncytial blastoderm, which means one big cytoplasm containing so many nuclei at the same stage. Now, advantage to fly is that all this bicoid protein, which is translated in tons present in the cytoplasm, this is the point where this is the source where messenger RNA is being translated, but it's a big cytoplasm. Now it diffuses freely. Okay. Same thing happens on this side. Same thing happens on this side. Sorry. Oh, ये क्या हो गया? Do you still see my screen or no?
No, sir.
Why it went? Abdullah, सारा तेरा कसूर है. So let's take this color now. Do you see my screen now?
Yes, sir.
What's wrong? Want to keep your ink annotation? Keep. Yeah. And I ask you to go to this. Yeah, but why you become red? Huh? I want you to remain blue.
सर राइट क्लिक करें टैबलेट पे, वो कोई कलर चेंज होता है, डॉक्टर फैसल करते होते हैं
यार.
टैबलेट पेन का ही चेंज हो जाता है कलर.
अच्छा, let me, मैं यहाँ पे blue कर लेता हूँ यार. So on this side, Nano's caudal messenger RNAs will be expressed. Since it's the same cytoplasm, what we will have a gradient. Nanos and caudal gradient from posterior to anterior. Do you see my screen? And
yes, sir.
From anterior to posterior, we have bicoid gradient. And in between, why? Let's go. Yeah. In between now, what we have, so let me. So it's not allowing me to work there. Let's draw embryo in the background. So this is the embryo, and these are the two gradients. Here blue one, here red one or pink one. Okay. Oh, sorry. Go back. Yeah. So अब्दुल्ला, तू मेरे से टच करा ले टैबलेट को.
क्या हुआ सर?
वो खुद ही अब कुछ करता जा रही है टैबलेट. ओके, फिर वही काम. चले. Anyway, so when we have so many cells, because nuclei तो divide हो रहे हैं ना? Is it right? Nuclei keep dividing, and these gradients are there. So cells which are exposed to highest concentration of bicoid, let's draw cells here. The cells exposed to maximum concentration of bicoid will start expressing different genes. Then this threshold gradient, this, तो एक फ्रेंच फ्लैग मॉडल आपका सेल्स की फेट, सेल्स की आइडेंटिटी डिफरेंट एस्टेब्लिश होना शुरू हो जाएगी. This is a nice picture. So maternal bicoid messenger RNA is present in abundant. This is the experimentally they have shown. If we draw X on X-axis, anterior posterior axis, and on the Y-axis we have the concentration of bicoid protein and messenger RNA. So this is the messenger RNA. Look, just in the 1/10 of embryo on the anterior side it is present, but when protein gets translated, messenger RNA gets translated, look, you have much more protein and it's a gradient, highest protein then low protein. Another maternal effect gene, hunchback, it does not give us a concentration gradient, rather it is present throughout the embryo from anterior to posterior, but there is a protein on the posterior side. I told you here messenger RNA of nanos is present, a posterior group maternal effect gene. When it gets translated, its protein is present here and it blocks the spread of hunchback. Now you have a gradient of nanos. So these opposing gradients, when you have few cells within the embryo, so the cells which are exposed to morphogen gradients, posterior morphogen gradient, anterior morphogen gradient, etc., their fates are becoming different, or future fates are becoming different, although they haven't yet committed to anything. Now, what happens that, I forgot to tell you that these maternal effect genes called bicoid, hunchback, nanos, caudal, etc., then 9 to 10 experimentally very crucial maternal effect genes shown in fly system. There are many others as well, but the ones which we routinely learn in the bio is these are transcription factors. Transcription factors. Now I told you it's a one big cytoplasm. Each nucleus is receiving one or the other morphogen gradient and receiving a specific concentration of gradient, these proteins. But these transcription factors are transient. They disappear after 30 minutes. But the changes they have induced here in the nuclei, they are kind of, if it's a normal development, they're kind of linear. They have to take to now next level. And once bicoid, hunchback, these maternal effect genes, they disappear after giving you two big gradients and dividing the embryo into two big regions, anterior gradient, posterior gradient. Before they disappear, they activate, because these are all transcription factors, they activate certain set of genes within the nuclei within the nuclei which are present in this embryo at very early stage, first 30 minutes to 1 hour or so. That nuclei, based on the concentration gradient of bicoid or concentration gradient of nanos, etc., the nuclei will activate certain genes which we call gap genes. Gap genes are also transient, and these genes are also transcription factors. Why we call them gap genes? Because if they are mutated, if you lose proper expression of gap genes, for example, Krupel is a gap gene, NERPS is a gap gene. If you lose expression of Krupel, NERPS, or other gap genes, embryo loses big gap. Like this particular region will be totally absent, gone. Or maybe this one will be gone. That's why they are called gap genes. Big gaps in the embryo are seen. This is expression pattern of gap genes, Now, gap genes, I told you, are also transient. They disappear. But before they go, they activate another set of genes in these nuclei. So gap gene transcripts and proteins, they bind to the nuclei. They activate certain sets of genes which are called pair rule genes. Pair rule genes are also transcription factors, but they are very interesting ones. They are interesting ones because, you know, they are expressed in alternate stripes. At this stage, you can see 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, and the first one will be here, 14. Did not we talk about 14 parasegments? It gets stuck. Did we talk about here 14 parasegments? So when we say that pair rule genes are expressed in alternate stripes, actually each pair or the two major pair rule genes which we teach or we learn during development is even skipped and Fushi-Tarase. Even skipped, if you stain using antibody against and look under microscope, you will find even skipped expressed in all the odd number. Sorry, if this is 1, 2, 3, so even skipped will be expressed in 1, 3, 5, 7, 9, 11, 13, blah. And Fushi-Tarazu will be expressed in the even segments. And these 14 stripes, they tell us the earliest segmented body plan is laid down. Now, these pair rule genes are also transient. After some time, they disappear. But before they disappear, they activate key genes, which are called segmentation genes or segment polarity genes. These segment polarity genes, and by this time, when segmentation genes are active, embryo has become cellular blastoderm. Before this, it was syncytial blastoderm. Now, cells are individual cells. Each cell has its own cell membrane, and segment polarity genes, many of them are cell signaling genes. Look, the intelligence or the intelligence behind this developmental plan. When cells are individualized or cell membranes are made around each nucleus, now simple diffusion will not take place. Now the communication along anterior to posterior will only be possible through cell membrane signal transduction, activating certain genes within the nucleus, and then cell-to-cell communication. Okay. Now, at this stage, when different segments are established, embryo has attained, the cells within the embryo has attained certain fates are determined already. I told you these first 3 segments are going to be head region, the next 3 thorax, and then next 8 abdomen. The segment polarity genes, they actually give polarity within the segment. Now let's say you have a segment within the embryo. Each segment will be having anterior and posterior compartment, and there are the cells. Now each segment has many cells, hundreds of cells. What is going to happen that there is an unknown boundary. We call it compartmental boundary. Cells from this compartment within the same segment are not going to mix with the neighboring cells. There is no China wall, there is no physical wall, but let's say these 2 cells are lying at the compartmental boundary. This cell is never going to mix up with this, but they're communicating with each other, they're telling each other. And then the distant cells away here are also, when further development will take place, they will contribute to tissue and organ of the same fate, but the pattern as a result of morphogenesis, as a result of pattern formation, will result in different. Just like you had here, we learned about the sonic hedgehog. You know, the cells within the same segment, they are determined to make hand and the different parts of the hand. However, they are going to contribute to different organ, thumb, etc. Similar kind of things are happening in flies. And segment polarity genes, they activate the genes which we know as Hox genes. I told you Hox genes are called master regulators of development. And they are the ones which give specific identity to different cell types. They lock specific identity of a cell type. And then, of course, differentiation takes place. If you look at gene level, these are the Hox genes, master regulators of development. I told you the lessons we learned from flies, they hold true in humans as well. And this slide may shock you. This may be shocking. The organization of the Hox genes in fly, UBX, ABD, ABDB, Antennapedias, sexcombs reduced, deformed, Proboscipedia, and labial, they come in two big clusters. On chromosome 3 in fly, one region is called bithorax complex, the other one is called Antennapedia complex. Now, early on, look at the expression pattern. Labial is expressed in head region, Proboscipedia, deformed here, and then you keep going. Antennapedia complex genes are expressed in the first 4 parasegments, while parasegment 5 to 14 you have these bithorax complex. UBX is expressed in very specific segment, then ABDA, and then ABDB. So, for example, the ABDB is expressed in the most posterior part of the embryo, and the labial is expressed in the most anterior. So when we say why this is so important, the genes, the expression pattern, so the genes which are expressed early on in embryo, we learned how they are expressed early on. They are expressed here. They are maintained in the characteristic cell type specific manner by the Hox genes. And Hox genes are these genes. They are maintaining the early patterns. And through mitosis, when adult develops, you see the color code? Through mitosis and even in the differentiation, when eventually differentiated, terminal differentiation takes place, from embryo all the way to terminal differentiation, these gene expression patterns, they are maintained, okay? And if you look at Drosophila embryo, early development and all the way to adult, and now look at your development, mouse development or human development, the Hox genes, this is the Antennapedia complex, and this is the bithorax complex. Their expression, this is the mouse embryo, their expression pattern is exactly identical. This is what we learned in flies, and what we learned about sonic hedgehog, digits, etc. All this have fundamental basis in fruit flies. Now, I told you these are master regulators of development. They're so crucial. And why they are master regulators of development? I told you misexpression of one key gene may lead to death, may lead to severe deformities in our development. This is the normal head, okay? But look, you have the legs now. Look, normal legs developing in the head region. This is just due to misexpression of antennapedia. Antennapedia is supposed to be expressed only in the legs. Antennapedia gives leg identity. Leg specific cells express this. Expression of antennapedia causes leg specific identity or fate determination and eventual differentiation. Here in this region, antennapedia is supposed to be off. It is supposed to be on only in legs. If you ectopically, ectopic means away from its natural location, if through mutagenesis, antennapedia becomes on here, you will get legs on the head. We call such mutations homeotic transformation. Homeotic. Homeotic, the word is from homeosis. One body part takes up identity of another body part. It's called homeotic transformation. Another gene which is not in the which is not present in the bithorax complex or antennapedia complex, but is known as a master regulator of development, is ILS. ILS is responsible for normal eye development. If you express ILS in legs or wings, look, you develop eyes on the legs or eyes on the wings, eyes on abdomen. So this development is so crucial. You cannot mess up with the developmental plan in the differential gene expression, which is established in a very, very specific developmental plan. Through developmental plan, gradually cell fates are established. If you mess up with that, you will die. You will develop severe developmental abnormalities, severe morphological phenotypes. Now, what is specific about Hox genes? Hox genes are basically transcription factors. They contain 180 base pair DNA sequence. Let's say if this is a Hox gene, okay, and there is a specific 180 base pair sequence. When their transcription takes place, eventually protein is made. This protein contains 60 amino acids. 180 base pair corresponds to 60 amino acids, which constitute a domain which is called homeodomain. And homeodomain in DNA is called homeobox. Homeodomain binds specific sequence. So these transcription factors, they go and bind in chromosome wherever you find this homeobox. Specific DNA sequence in the promoter region of key developmental genes and kick them on or off. And they are conserved in mammals and in plants. This neck shock. So you just learned, you know, we have flies, invertebrates, we have mouse, humans, vertebrates. We have plants also containing Hox genes. Plants suffer similarly as animals suffer due to misregulation of Hox genes. Oh, here are the plants. What is the function of Hox genes in plants? Plants also have symmetry. You know, you have sepals, petals, stamens, carpel. And it's a symmetry. Sepals, petals, stamens, carpel, carpel. You go from here to there. Stamen, look, carpel, stamen, petals, sepals. It's a symmetry, no? This symmetry is achieved by these Hox genes. And what happens if you misregulate Hox genes? Look, petals are gone. Petals are converted into sepals. Or you have many carpels. Or you have many petals. So this pattern formation in flowering is basically due to this Madspox genes, which in mammals or in flies, invertebrates, we call them Hox genes. Okay. There is specific gene expression pattern. So these genes in plants, the ones Elliot Meyerowitz lab discovered that they had A, B, C model, A, B, C, A class genes, B class genes, and C class genes. Just looking at the phenotype of the plants, mutant plants, they said, okay, we have phenotype in which, let's say, we have, duplication of carpels. Many carpels are there, and due to duplication of carpels, we don't see petals at all, or we have stamens, or we have, duplication of sepals or duplication of petals. So they, based on phenotype, they hypothesized we have 3 classes of genes, A, B, and C, and they generated this ABC model of flowering. I would like you to go and read this. This is amazing discovery, one of the finest discoveries in developmental biology. Now, let's take 5 minutes break here, because we are going to move into molecular side. We are going to move into epigenetics. Geospa, beta.
Sir, I had a question. What's the difference between genes that are recessive and genes that are switched off?
Genes which are recessive and genes which are switched off. A wonderful question. Remember, a dominant and a recessive, these are not genes. Never ever confuse dominant and recessive with genes. These are phenotypes. Even people, when they go to PhD level, they do this mistake. Fundamentally, the right concept is it's the phenotype, which is dominant or recessive. Genes are not dominant or recessive. However, once you discover that dominant, let's say, dominant phenotype, let's say this leg on head, hypothetically, this is due to antennapedia gene, okay? You will not say this antennapedia or antennapedia mutation is dominant. No. You will say antennapedia phenotype I have discovered is dominant. It is due to mutation in antennapedia, and allele of antennapedia, let's say I have antennapedia allele 1, is phenotype of antennapedia is allele 1 is dominant. However, you may discover another phenotype, which is leg on head, antennapedia phenotype. Let's call it antennapedia phenotype. But this turns out to be, it requires minus, and you will say antennapedia 2 shows me recessive phenotype. You may discover that antennapedia 3 allele, antennapedia 4 allele, different mutants you discover, they all are dominant phenotypes. So dominant or recessive is used for phenotypes, not for genes. Alleles can be showing recessive or dominant phenotype, and then this will show you the second allele, which is recessive phenotype. It means it's not a strong phenotype. It's a very weak phenotype. Maybe one amino acid is changed to another. There's no complete loss of function, but these are complete loss of functions, the one which showed dominant phenotypes. Did I answer your question?
Yes, sir. Thank you so much.
So now we are going to look at the molecules and the processes which result in such differential gene expression or regulated transcription in eukaryotes. And since we say, cell divides into 2 and then, it goes 4, this is the fertilized egg, that's how cells keep increasing. Each cell remain diploid contain same set of genes. Question arises how they become different. We learned they become different because they express genes differentially due to differential gene expression. But then the next question arises how this differential gene expression is established and then maintained. If let's say Antennapedia gene, we just talked about this. If Antennapedia gene is present in the, this is the leg region, and let's say in the head region, Antennapedia gene is off, how through mitosis, successive mitosis, it remains on in the head region, off in the head region, but in a different place, it remains on. What you have actually created, you have created a mutant-like situation. When you lose a gene function, here we just talked about losing a gene function. So turning gene off means you have kind of lost its function in head region, but you have kept its function through switching on gene in the leg region. But this losing function is not permanently lost, it's not irreversible. The genetic sequence, the DNA sequence of Antennapedia is 100% same in both the leg and the head region, the cell types, but only at a level above, at the epigenetic level, by regulating the promoter region. You know, here the transcription is taking place, but here it is repressed, switched off. So we call such a phenomenon epigenetics because the underlying sequence remains the same, but the gene expression is changed by acting one level above the genes, and that is that gene expression state off or on is also heritable through mitosis. So how transcription in eukaryotes takes place, I'm not going to go through transcription. I already told you, we learned this in great detail, that DNA is packaged in a nucleosomal form, and eventually in chromosomes you have DNA present as nucleosomes, and then this DNA is transcribed. The messenger RNA encoding genes are transcribed by RNA pol2 in eukaryotes. RNA pol1 transcribes the ribosomal RNA, and RNA pol3 transcribes the tRNAs. Okay, there's division of labor because there are 20,000 genes in eukaryote, in for example in humans, or 15,000 or more in fruit flies. So many genes are to be transcribed, so there are different kinds of polymerases: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. We will particularly stick to the processes which are being led by RNA polymerase 2. It binds to gene-specific promoter, and these promoter elements also attract transcription factors, and then normally genes are turned on. Messenger RNA is synthesized, messenger RNA is exported after splicing, and in the cytoplasm we have translation by the ribosome, and eventually we have proteins. Okay, this is what the general central dogma is. Now, transcription unit in eukaryote, we also learned about this. We have the promoter sequence, we have the gene sequence. Gene sequence comprises of introns and exons. Then we have the termination sequences where RNA polymerase falls off. And then we have the regulatory elements in addition to promoter elements in the DNA sequence. So let's say this is the promoter element, data box, etc. Then this is the gene sequences. This is place where RNA pol2 binds. But you know, you may have other DNA sequences which we call regulatory sequences. They can be upstream or downstream of the gene, and they positively or negatively impact RNA pol2 machinery and then contribute to either activation or repression. Okay. Now, transcription machinery, we already talked about RNA pol2, general transcription factors, etc. Data box, TF2D, general transcription factors. These are basically proteins other than RNA pol2 which recognize Tata box or bind to the promoter elements together with other proteins, which we call general transcription factor. So here is a cartoon which shows, the promoter element, which is bound by TF2D protein, and then it recruits other transcription factors. RNA pol2 comes in. General transcription factors on RNA polymerase bind the promoter element. Then comes the enhancer element. Enhancer is basically, these are DNA sequences, a signature sequence, which is also regulatory element, and it positively impact this RNA pol2 transcription factors, etc. And this complex then leads to transcription, messenger RNA synthesis. Now, enhancers, they attract proteins which are called activators. Activators from the word activation, they positively impact the RNA pol2 to help RNA polymerase 2 in ultimate transcription or messenger RNA synthesis. There can be repressors as well on certain regulatory sequences. In contrast to activators, we may have a repressor protein bound here or here in the promoter region, which blocks, which have a negative effect on the transcription machinery and does not allow the transcription. And in that case, transcription is halted or gene is turned off. Repressed is the right word. Now, this here is shown, like a linear thing. This is easiest way of teaching so that you can visualize things. But normally these are long range, long distance interactions. You may have 1 million base pairs between a promoter and the enhancer. 1 million base pairs far away from the gene, upstream of the promoter or downstream of the promoter. And the chromosome, within the chromosome you have such an interaction. This is where your gene is. This is where the promoter element is. This is where RNA pol2 is found. But this 1 million base pairs looped around, and you have this activator brought closer to the promoter element, and then it will have positive or negative effect. That's how the activated or repressed transcription takes place due to these regulatory sequences in the eukaryotes. Now, let's talk about, that was general transcription. That was how transcription process is taking place. Where does the epigenetic comes in? We said a level above the DNA sequence. We said if a gene sequence is A, C, A, C, G, A, C, G, A, T, A, T, A, Tata box, blah, 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 A, T, A, C, C, A, T, A, U, G, blah, 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 blah. And then you have the transcription termination sequence. This is the sequence of one gene. The same gene, I have A, C, G, A, C, G, T, A, T, A, T, A, Tata box, then T, A, C, C, A, T, A, U, G, transcription termination sequence. DNA sequence, same gene. Let's say now this one is present. This one is present in cell type A, and this one is present in cell type B. This gene should be on in cell type A, but should be off in cell type B. How cell is going to do this? One possibility is we cause a mutation and permanently kill this gene. In B, we will lose function and keep it intact in A. But this is very dangerous. Nothing in nature is junk. You may require this gene during subsequent development of A later on or B later on. what nature has invented is a very useful chemical modification that is called 5-methyl cytosine. What system does, it says, okay, I need to switch off gene here in this cell type. Okay, I'll add a methyl group on the cytosines. It looks like this. This is a cytosine. A methyl group is added on the 5th position. We call this 5-methyl cytosine. We also write it like this, 5-methyl cytosine, or it can also be normally on the top of C, just to show cytosine is getting methylated. Wherever you will have in the promoter regions, if you have methylation, no matter how strong promoters are, gene is off. What has happened? Gene sequence is 100% same. In all cell types in my body, this gene contains same gene sequence, but it got methylated in cell type B, and due to methylation, RNA polymerase 2 cannot bind, transcription factors cannot bind, activators cannot act, and the net result is repression, gene silencing. Okay, in the promoter region. Now, it doesn't stop there. I turned it off, methylation on cytosine, and look, beauty is the system is very specific. It's not random. It's called symmetrical DNA methylation. There can be asymmetrical as well. Symmetrical means CG in one strand, the opposite strand will also have CG. You go 5 to 3 and 5 to 3 orientation. Here also CG. It was CG. Okay. So symmetrical methylation is maintained. So now this cell is going to divide into 2 cells. You know how DNA replicates? Conservative. 2 mother strands separate. One of the strand will remain methylated because this is a covalent modification, very stable modification of cytosine. It remains, and the new strand, which is synthesized, is unmethylated. This state is called hemimethylation. Half of the DNA is methylated. This hemimethylated state, right at the replication fork, where this replication is taking place, is recognized by an enzyme called DNA maintenance methyl transferase. Okay, and it methylated newly synthesized strand. What is the net result? The mother cell where gene was off, it divided into 2. The gene will remain off here as well. However, in cell type A, where it is not methylated, where it is required, it will remain on. When this A will divide into 2, gene will be passed on in the on state. So here we have now 2 cell types. Gene A contains gene in the on state. Cell type A keeps gene in the on state. Cell type B keeps the gene in the off state due to DNA methylation, which is maintained by maintenance methyl transferase. The enzymes which catalyze this methylation de novo means from scratch on the naked DNA. यहाँ पे कोई signal नहीं है, अचानक signal आया है, इसको off करें. Methyl transferase, DNA methyl transferase is the enzyme which methylates, and then subsequently this methylation is maintained. Okay, what is the net effect? Net effect is DNA sequence in A cell type and B cell type. Primary DNA nucleotide sequence is same in both the cell types. In one, it has become off. In another, it has remained on. In one where it has lost its gene expression is due to this epigenetic modification. We call this epigenetic epi means above. And genetic, you all know genes. You know what genetics is. Study of genes, inheritance of genes. So above basic genetic code, which is the DNA, you have introduced this epigenetic modification, which is a chemical modification, covalent modification, and this is heritable through mitosis. From 1 cell to 2, 2 to 4, 4 to 8. So this fulfills the criteria of genetics. Inheritance is taking place. In 1960s, they discovered that, the other component of chromosomes or chromatin, we learned there are 4 different histone, H2, H2B, H3, H4, and 2 molecules each are present in a nucleosome. They discovered the amino acids in histone proteins. If we stretch, imagine we have linearized the protein sequence of histone H3. Amino acid, arginine 2, amino acid 1, here will be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. What they discovered that the amino acids, let's say arginine 2, lysine 4, lysine 9, this is the position of amino acid within the protein. 10th position amino acid, which is serine, they can be covalently modified as well. Methylation, 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 phosphorylation, acetylation. Methylation, acetylation, acetylation, methylation. So you have, we call it histone methylation. What we just learned on the previous slide was DNA methylation. Acetylation, phosphorylation, ubiquitination, and you keep going like this. What they discovered that, different amino acids are modified, but they did not really understand the role of these chemical modifications in the chromosome. They thought they maybe are required for packaging, because nucleosomes are normally packaging proteins. In late 80s and early 90s, they discovered if histones are acetylated. Look, histone H4, lysine 5, lysine 8, lysine 12, lysine 6. They discovered that these 4 acetylation states, and they said acetylation generally is whenever nucleosomes are acetylated, the gene is active. Gene is not off, which means deacetylation state, when acetylation is absent, those nucleosomes are associated with genes which are turned off. And they correlated in humans, in mouse, and flies that chromatin, where nucleosomes are highly acetylated, they correlate with genes which are on. And genes which are off, for example, which are DNA methylated, they will, these ones, all the genes which are silent due to DNA methylation, they correlate, they are, the ones which are demethylated, they are deacetylated. They lack acetylation. Okay. So in early 2000, they discovered in this century, they discovered that methylation of histones is also very important. Although methylation of histones was known in early 60s, okay, but they only discovered the function that DNA, when DNA gets methylated, so 5-methylcytosine in plants and in animals, mouse and humans, etc. Wherever DNA will be methylated, histone H3 lysine 9, lysine will be methylated as well. So which means covalent modification of histone H3 lysine 9 correlates with covalent modification of cytosine and chromatin. And together they contribute to repressed or silent genes And once differential gene expression is established, off genes can be inherited through mitosis through these covalent modifications. However, they also discovered that lysine 4, when it is methylated, it antagonizes lysine 9 methylation. So a gene can be packaged in nucleosomes, either lysine 9 or lysine 4. And they said lysine 4 methylation correlates with acetylation. And so lysine 4 AH3, histone AH3 lysine 4 methylation plus H4 acetylation, these are marks for active chromatin, and they are also mitotically inherited. These are epigenetic modifications. Underlying DNA sequence, I told you, underlying DNA sequence remains same, but the nucleosomes in which they are packaged, they are modified covalently. And that's how genes are switched on, switched off, maintained in the on state, maintained in the repressed state, and through mitosis. During development, this differential gene expression is inherited. So these are basically, DNA methylation, histone modifications, how they translate into the on or off state. For example, I just draw here. This is the cytosine methylation. It recognizes, it's methylated now. CG. Let's say this is promoter region where cytosines are methylated. Now, this comes under, this DNA is packaged. Let's say this is DNA which is on this nucleosome. And here we have, histone H2A, H2B, H3, H4, and we have these N-terminal tails. I forgot to tell you, these are called N-terminal tails, are non-structured basically in histones. There is globular core histone, but the N-terminal is like non-structured. We call them N-terminal tails. Now, if we have methylation, okay, methylation is recognized. This methyl mark is recognized by specialized proteins, which is called methyl binding domains containing proteins, MBDs. Short form is MBDs. And then these MBDs, they recruit HDACs. HDACs are histone deacetylases. Histone deacetylases remove these acetyl marks. So what are you doing? You are preparing this gene, this specific gene, to go in the off state. And histone H3 tail, which carries lysine 9, what they say, I mean, in mammals in particular, they say it gets trimethylated. So 3 methyl marks come here. This is the early, they say it's a sign for repression or gene silencing. This is going to dictate or send a signal for DNA methylation. Cytosines will get methylated. Then HDACs will come, MBDs together with HDACs will come. DNA and the histones H4 will be deacetylated and gene is turned on. And reverse events will take place when you have to switch on a gene from off to on, or if you have to maintain gene in the on state. You can imagine if you have to maintain gene in the on state, then in the lysine 4, you will have lysine 4 of histone H3 will be trimethylated, and lysine 5, 8, 12, 16 of H4 will be acetylated, and you will not recruit MBDs, or methylation will not come, and gene will be turned on and remain on. What are the examples? I just want to give you example. What time I have? I have short time. The biggest example at your level is the X chromosome. So in human females, we have 2 X chromosomes. In males, we have 1 X chromosome. So there's unequal dosage. So it means male will express 50% of the genes on X. In females, we have 2 copies, 50 coming from here, 50 coming. So expression level is different. What nature has evolved, it has inactivated. Despite there are 2 X chromosomes in females and in males 1, in order to be equal dosage for each gene which is on the X, one of the X chromosome in human females remains inactive. We call this phenomenon X inactivation. This is beautiful phenomenon. And this X inactivation results in equal dosage of genes which are on X. Now, in us, males and female, gene dosage is equal. How this is achieved early on? Here you can see this tiny dot. This is very early development. This is one of the X's converted into Barr body, after the name of the scientist Barr, who discovered this. So what they discovered that in next 50 years, what we have learned that, you know, DNA methylation of the chromosome, which is to be inactivated, lysine 9 methylation, lysine 27. I did not talk in detail. You know, we talked about Hox genes. Hox genes, if they are repressed, they do not contain lysine 9, but lysine 27 trimethylation. This is very important in development. From developmental point of view, lysine 27 trimethylation associated with repression of gene silencing is a key mark for Hox gene repression. So X chromosome also get lysine trimethylation. And the proteins which recognize lysine 9 or lysine 27 trimethylation, they are different complexes, but they are conserved from flies to mammals, okay, and in plants as well. And there's a non-coding RNA called ZIST as well. ZIST is the earliest signal, the chromosome which is to be inactivated, which is to be shut down here, and that starts expressing inactive X chromosome. It binds onto the chromosome which is going to be inactivated, and then DNA methylation, histone methylation, etc. comes in. It's, you know, crumbled, jumbled, and the other chromosome which has to remain active, it remains in a normal structure and genes there on. Genomic imprinting is another phenomenon where sometimes, not sometimes, always all of us inherit 200 or so genes from our parents in certain state of gene expression. For example, we receive IGF2 and H19. These two are neighboring genes. Okay. I forgot now, and I always forget this. Let's say gene A, we name IGF2A and B, whatever. We inherit them in such a way that only mother's copy, maternal copy, is active and paternal copy in us is off. Always, in each one of us, there will be another gene where a totally different gene, maternal gene copy is off and father's copy is on. We call this parent of origin gene expression. Only one copy, or also we call them monoallelic gene, because only one copy, one of the two copies are expressed in our normal body, all individual, all humans, and they will be conserved. Those genes, either it will be mother or father's. It's not random. This phenomenon is called genomic imprinting, and this is from parents to offspring, which means through meiosis. It's not mitotic inheritance, and that also involves DNA methylation and histone modification. Okay. How do we, you know, I think we have learned enough of the coordinated gene expression or regulated gene expression we just talked about. This is another example. What I want to show you, all what we have learned about development. starting from key processes and stages, which was cleavage division, blastocyst, gastrulation, neurulation, organogenesis, etc. We have learned in some of these model systems like Xenopus, chick, etc. Again, then the epigenetic modifications and the epigenetic gene regulation, which we have learned, DNA methylation, histone modifications, which play crucial role in differential gene expression, establishment of differential gene expression and maintenance of gene expression, and consequent cell-fate determination and differentiation is also learned in these model systems. For example, in flies, we have very little DNA methylation, early developmental stage. So all we know about developmental DNA methylation, actually plants contributed so much to our information about DNA methylation and its role in gene silencing. And then the mouse systems and the hen or the chicks or the Xenopus also contribute, but the enormous amount of DNA methylation associated with gene silencing and development came from plants and mouse. So I think this week talked yesterday already, Spielman's organizers and developmental organizers. And so with that, I would like to thank all of you and would like to know if you have any questions, please feel free to go ahead. Any questions? So if we have no more questions, thank you very much. If you have any questions, you can approach me later on as well. Feel free to write me an email. Abdullah is there. You can ask Abdullah whatever questions you have. Your exam regarding this module will contain, few genetic problems, and then you will be having MCQs as well. Okay. Thank you very much. I wish you a good evening. Allah Hafiz.
Allah Hafiz, sir.